Blood transfusion has become an increasingly vital and sometimes life-saving procedure in veterinary medicine, particularly for our canine companions. Much like in human medicine, the ability to store blood in blood banks for extended periods has revolutionized transfusion practices, allowing for readily available blood products for up to 42 days in both humans and dogs. This prolonged preservation is made possible by storage solutions and specific temperature controls, typically around 4 degrees Celsius, plus or minus 2 degrees. However, this seemingly beneficial storage period comes with its own set of challenges. During refrigerated storage, red blood cells undergo a series of biochemical and morphological alterations collectively known as storage lesions. These lesions can potentially lead to adverse reactions in the recipient after transfusion. Scientists categorize these changes as biochemical, biomechanical, or structural, all of which can contribute to immunomodulatory effects, thereby impacting the safety and effectiveness of the transfusion. These changes include alterations in energy metabolism, an increase in oxidative damage, impairments in the red blood cell's ability to deform, the formation of vesicles, modifications in the phospholipid structure, and changes in the sodium-potassium gradient across the cell membrane. The accumulation of these alterations in the blood storage bag can also lead to an increased rate of red blood cell destruction, known as hemolysis. Research in human transfusion medicine has indicated that certain components derived from white blood cells, such as pro-inflammatory mediators like lipid peroxidation products, can influence biological responses after transfusion. These responses could potentially contribute to undesirable transfusion-related complications, such as transfusion-mediated immunomodulation or a serious condition known as transfusion-related acute lung injury, which is a form of acute respiratory distress syndrome. Given the well-established benefits of leukoreduction, a process that removes white blood cells, in reducing the occurrence and severity of storage lesions and adverse events following transfusion in humans, it might seem logical to apply this technique universally. However, the practice of performing leukoreduction on human and canine blood units before storage is not yet a standard procedure worldwide. This study offers compelling molecular evidence highlighting the substantial impact of leukoreduction on the metabolic landscape of canine-packed red blood cells during refrigerated storage. These findings not only expand upon existing knowledge from human studies, but also provide valuable insights for future research aimed at developing improved storage strategies for canine blood products, potentially through the design and testing of novel storage additives.